I will get started. Good morning, everyone, and thanks for coming to the day 2 of our Precision Medicine Research Symposium, which is a joint effort between Cornell Ithaca, Weill Cornell, as well as Cornell Tech. To my knowledge, this is one of the early or the first to bring all 3 campuses together to organize something, and it's really a major initiative that is coming out of thoughts across the 3 campuses and intercampus bridging opportunities that were created over the last couple of years by the leadership at Cornell, including Gary Kureski, who has been leading some of these big efforts in the area of bridging the gaps, as well as Weill Cornell Qatar. Yes, and Weill Cornell Qatar. Sorry, so 4 campuses altogether. So definitely the first one then. So so great opportunities over here to interact with people. We gave a little bit of introduction yesterday, all the organizing team members, Olivia Alimento, Cora Sternberg, myself, and Tanzim Chaudhry. Yesterday about how various campuses are bringing things together. We had a wonderful day 1 with 2 major sessions that we had. One was around personalized approach to patient care, which had an opening talk by David Danus and had some great insight into how precision medicine can make a difference in cancers. We had some exciting talks from Lisa Newman about disparity in diversity, socioeconomic diversity, as well as racial diversity research related to breast cancer and other topics. Many other talks that we had that were really exciting. Betsy Ross presented on the neurological diagnosis, and there was some interesting work on Alzheimer prevention from Richard Isaacson. Then we had a second session yesterday, which focused on innovations in healthcare technology. And it was particularly very exciting to see how various technological side of things were integrating with the precision medicine side. We had Sensing to Intervention Loop for Health talk by Tanzim Chaudhry, and then very exciting work on mixed reality, where you can play around with things by putting some interesting tools that's developed along these lines, and deep learning for assessment by Iman Hezrosalia. We had a poster session also yesterday. Some very good discussions were there, so overall, I think day 1 was quite successful, as we see, and it allowed a lot of interactive opportunities between people. Today we have another packed day, and our first session is from bench to bedside, accelerating clinical research. It has experts from Qatar, from Ithaca, and from Weill Cornell. And it's, one thing that I didn't say yesterday, Cornell Ithaca, as some of you may know, offers various different levels of or sides of interaction that maybe you should interact or capitalize on. So there is biomedical that is into several departments, including biomedical engineering, biomedical sciences, mechanical, chemical. Pretty much every department has some level of foot into bioengineering, biomedical side of things. And then you have the vet school side, which is doing amazing research on where you can look into various animal models. I was having this great discussion yesterday about biobanking that could be work together with the biobanking at Englander Institute, and so a lot of opportunities that you can take a look at. So some of the talks today are going to also touch on those topics, and you will see some of the integration. Later in the day, we have 2 panels one on digital technologies for clinical care, and then panel 2 is on translational research together. So I'm looking forward to some exciting discussion over there. Then later, we have working groups that are along liquid biopsies, artificial intelligence for cancer, engineered ex vivo models, organoid platforms, precision medicine engagement and education working group as well. and a lot of interactions throughout the day. So without further delay, I would like to start our bench to bedside session, and our first speaker is Dr. Karsten Schuhe from, did I say it right? Yeah. Close, okay. From our Qatar site, and okay, let's get to go.
All right, thank you very much. So thank you, Olivier, for inviting me here, and it's really a great pleasure to be here. So let me just get into it right away. So I really don't know what the audience is here. I have a lot of slides, but I've seen the other guys of you also just showed teasers, so come back to me if you want more details. It's more like a quicker slideshow. So this is my conflict of interest slide. Yeah, Olivier already mentioned that, but I think I still remind you once again. So I come from the Qatar campus, so that looks like that. So that's Weill Cornell in Qatar. It's fully a branch campus here from Cornell. I don't know whether it's really clear to everyone. There's lots of research going on there. I'm personally affiliated with the Department of Harold Weinstein and also with the Englander Institute. So the questions we address in my team and with colleagues around the world is actually to study what makes us different, our individuality. And I think I don't have to go deeply into this, that there are all these different lifestyle factors that play a role, whether you're physically active, the drugs you take, the gut microbiome becomes a big topic. And then something we worked a lot on is the genome, how the genome impacts our health. And then how does it impact, and these are a bit the diseases we are most interested in my team. It's like in Qatar we have a lot of diabetes, cardiovascular disease, cancer, and Alzheimer's is something we do here with my team. Also Jan Krumzik participates in the Alzheimer research here in the Englander Institute. So what I think is a bit our strength is to do the multiomics, to have this as an intermediate phenotype. And we've learned a lot about genomics, and when people say multiomics, they actually think about genomics, maybe epigenetics, transcriptomics, but there's a lot more. I mean, you have the paradigm of biology, you go to proteins. Something I've worked a lot on is the metabolites, metabolomics, and then you also have like the side things like the epigenetics, microRNAs, protein modifications like glycomics and so on. So what we study a lot is to do this in population studies, and what epidemiologists do, they study samples from many, many people, and then they do associations between genetics and outcomes, so that would be genome-wide association studies. Epidemiological studies, how do these different lifestyle factors influence disease? But the point is that population-based studies, they identify risk factors, and they can teach you something, but they very rarely really reveal the causal pathways and give you a detailed understanding of the pathways. So the idea is now, how can we make that, bridge that gap between like the causal factors and the disease outcomes? And the idea is to put in the intermediate traits. So to say, we use the epigenomics, metabolomics, transcriptomics, and so on, deep molecular phenotypes, in order to actually reveal the affected pathways that then lead to disease. And then you can do GWAS with metabolomics, with proteomics. You can do proteome against disease, metabolome against disease, and so on. And that's, I want to show you a bit some of examples we did there. So the first thing we started off, I think about 10 years ago when I was still in Munich, was genome-wide association studies with metabolomics. And I think I want to start with that. But that gives you a bit the concept for all the other things that come. So what we very early on identified was what we now call the genetically influenced metabotype. And that's actually a genetic association with common genetic variants with metabolic outcomes. And what you find very often is that you have like a very strong association. Now, this is not monogenic disorders. These are just common variants, SNPs in the population. And in this showcase, the FADS1 gene, we find, for instance, that major allele homozygotes of one of these alleles gives you a 50% faster metabolic rate to create arachidonic acid, and that's a very important metabolite to create prostaglandins and in the inflammatory pathways. I think the concept here is, I think I like this kind of looking at these SNPs as valves, right? So in your body, and I think I have another picture here, so you would have like, I don't know, maybe every one of us has millions of different genetic variants in your body, and many of them, they don't do anything, but some of them, they act as these valves in your system. So they may throttle or increase the throughput of an enzyme, of a transporter, and so on. And then you would have people stratified by being fast, medium, and slow metabolizer of this enzymatic reaction. And then this is, of course, it's just one. So what we then thought is, are there more of them? And then, I mean, this is not disappointing, but I didn't know, I'm a physicist, so I didn't know Archibald Garrett at that time. So he was the inventor of the inborn errors of metabolism, and he did already 100 years ago actually predict that inborn errors of metabolism are just extreme examples of chemical behavior, which is probably everywhere. And the concept of, well, individual or personalized medicine, he came up with that already by saying that chemical individuality is actually a predisposing factor to disease. So I think the whole concept of precision medicine is, I think, goes back to 1909. So once we had these initial results from the GWAS, I said, now we want to map out the human metabolic individuality and then put up a roadmap to identify actually, well, all relevant. You can discuss what relevant means, but everything with a larger effect size and pathways that you can measure. And then what I will talk about in the end is how can we translate then this kind of knowledge of these different valves, if you want, valve settings in your body to biomedical application. All right, so what you see here is the metabolomics core facility of the Helmholtz Center in Munich. So that's something we built up in 2008 or so. So that's running already for 10 years now. And then we ran all kind of different metabolomics platforms, most of them commercial, but some of them also implemented in the core facility and measured metabolomics on big cohorts. So in Germany, there's a study called CORA. So that's the equivalent of Framingham. And now they move into the national cohort with a bit the equivalent of the All of Us study. So we use samples from these studies. And then over the next, well, maybe 6 years, we ran all kind of genome-wide association studies with different platforms and different techniques, NMR, mass spec, urine, blood, different cohorts. And I think the most impressive one, or the first really strong one with the company Metabolon, they have really very broad metabolomics panel. And what we identified there is what we then say is the human metabolic individuality, so what Archibald Garrett actually predicted. So each of these tiles here is one genetic variant in one specific enzyme. So you see up there FADS1, my example. So there are 37 that we identified at that time. So these would all be individual variants that would be different in each of you. So you could see there would be a combinatorics of 37 settings of valves in everybody in this body, in this room. And that would define your biochemical individuality, and by that, define your predisposition or protection from disease. And then the question is, how does that impact disease? So you can then go and overlay these variants with GWAS catalog. So all the genome-wide association studies that were run in parallel. I think all that started about 2006. So this is from 2011. So now there's more in the overlap. But what you see already, there's a strong enrichment in actually GWAS hits that overlap with metabolomics loci. And that means that these are really intermediate phenotypes to the disease, and also with risk, and some with just benign traits like body hate. And then, as time went on, we increased the power of the studies. Another milestone was when we had, this is now in 7,800 people, again with a Metabolon platform. So we measured, I think in that time, 400 different metabolites, did the GWAS, and what we found was 145 of these different genetically influenced metabotypes. And then you can put them into a network, and I think at that time Jan came up with an idea to also create Gaussian graphical correlation models between metabolites, and if you put that all together, you can create what we then called the Atlas of Human Metabolic Individuality. So you connect the metabolites here by their partial correlations, and then you link the genetic variants to them, and with that all together, you create a network where you can then say, I pull on certain metabotypes, on certain diseases, certain SNPs, and see what is the network around it. And I will show another example later on proteomics as well. So with that, I mean, that work is going on, but, well, for my team, we ran a bit out of samples. So Germany, we don't have these big cohorts. So now I'm collaborating with people in Cambridge. They have a study with 20,000 people. And that's really amazing. So what you see here is the Manhattan plots. So this is, on the one axis, you have the chromosome position. The other axis is the different metabolic pathways. So you have different amino acids and so on, and the p-values. And then each of these loci is a metabolic individuality phenotype, a GIM. Now, the thing is, when you go to 20,000, you can refine these associations with statistical power so that you don't only find one genetic association at that locus, but you can refine them into independent loci. And what you find in this example here is there's actually 4 different genetic variants that associate with about 15 or so different metabolites. If you look at the details, they're all related, unacylated amino acids. But the interesting thing is you find that some metabolites, so this is the effect, whether it is positively or negatively associated. So there's one major signal, so everything goes with this one genetic variant. And that might, for instance, be a variant that regulates the gene expression. But then you have others that are limited to rarer variants, also frequent ones. And some of them, like here the red one, and this one has a different sign here, have different opposite directionality. So what that means is you can now decipher the biochemistry that's happening at that locus, because this other variant, I mean, I don't have the details here, but you can look where it comes from. It could be a functional variant, changing the binding capacity of the enzyme, the specificity towards certain of these metabolites. So with all this knowledge, you can now go and dive into understanding what's going on there. And if you imagine that one of these is actually also overlapping with the cancer locus, for instance, you could understand a lot of what happens if a certain variant you find in a cancer mutation is overlapping with one of these enzymes. All right, so, and from there on, we thought about what can we do in terms of bringing it to the clinic. And I think one idea was to say, actually, these valves, they all make up drug targets. Because if you can change these valve settings, I could modulate your metabolism. So if I take the central enzymes where I know nature has shown me, if nature turns on these valves, because that's actually what the SNPs are, right, an experiment by nature. If nature turns that, and then I see the readout, which is the change in the metabolites, then it means that if I target that with a drug, the same reaction will come. It's the idea of Mendelian randomization, so there it does the same thing. So it means all of these variants or genes are actually drug targets where I know already what would happen if I targeted. So if I now can conceptualize that together and say, I have anything, let me just go quickly through that. Yeah, I mean, that's an idea initially developed with Professor Anna Halama from my group. So we got a grant from Qatar Foundation and just got a renewal on that one. So the idea would be, if you take a pathological state, that could be cancer, that could be anything else, if you then, with systems biology, identify the valves you would like to turn to actually adjust something, that could be metabolic co-lethality, anything, and then you would actually target these, you could re-engineer, actually metabolically engineer your pathways. And the idea is now to set up on this grant to take these 145 valves that we have. For many of them, we have already drug targets that exist for many of them, and then have a suite of bioinformatics tools that we developed here together with the England Institute, having metabolic assays. So we started on cell culture, but I think the organoids that you mentioned are extremely interesting to have things to actually prove that the thing is working. And then in parallel, you would need, of course, the metabolomics readout to monitor these things. So the idea is to build up a toolkit where you could metabolically engineer an organism, and that could be used for maybe penalizing cancers that have a weakness on glutaminase, for instance. We have some examples there. You could also use it maybe for diabetes. So I think there's a lot of things to be done there. But I think in the interest of time, I should maybe move on and say, so that was metabolomics. Now, we were lucky to actually have a SomaLogic platform. I think someone mentioned it yesterday. I think Jason did, right? So SomaLogic is a technology that allows you to measure protein concentration using aptamers. And we had a platform in Qatar for a few years where you could measure about 1,100 proteins in parallel, and we ran a GWAS, like the same concept with the metabolomics on them. And, well, the outcome was even more impressive. So this is the Manhattan plot from the GWAS we ran, and I think it was one of the first of that size. So what we identified is 451 independent, genetically influenced loci where the genes really are strongly associated with proteins. And then, of course, you can think about the same concept, overlay now these with all kind of other QTLs, like we can look at overlapping methylation QTLs, regulatory variants, EQTLs, transcript modifying variants. Then the proteins are also interesting. You have actually cis and trans associations. So the trans associations are extremely interesting because they link to protein-protein interactions. And then I think for clinical applications, in this case, we had 83 overlaps with GWAS hits on 42 different disease phenotypes. And that, I just want to quickly show you how that looks like. So that would be an association of a SNP with one protein, C7 from the complement system. Then you can grow that out with the GGMs, the partial Gaussian networks, and you see already things with make sense come up here, like other component system factors. Then you go to the GWAS catalog and say, this is a variant that goes with Crohn's disease, and there are actually 4 different genetic loci that overlap with our proteins. And then you see here all different kind of histories, and you see, like here, there are different ILs here and different ILs here. I'm not a specialist in Crohn's disease, but I think a specialist would now be able to actually link these together and get a better understanding of what's going on in these things. So as I said, we have 42 GWAS disease hits. So the network then looks like that, or like that. And that's extremely interesting. And I think yesterday we've seen the virtual reality things, and we're working with Olivier's lab here on actually making a visualization. I think Alex here? No, he's there. Okay, I think he can show you later on HoloLens application on these networks. So to actually try to, how do we disentangle these networks with virtual reality? At the moment, I do it in 2D. But even then, if I zoom in and say I grab, for instance, here I'm interested in Alzheimer's, I pull out Alzheimer's, and then I find what is associated with Alzheimer's. So the green ones would be the GWAS hits for Alzheimer's, here especially, of course, the ApoE variant. But the interesting thing is we also find a second one here. And I'm not saying all this is true. There are some caveats to the SOMA logic, but you can create things you can follow up on. So there is the classical GWAS hit for Alzheimer's, the ApoE association with ApoE circulating proteins we see, but we also find a negative association with RUX-F, the small nuclear ribosomal protein F. And the interesting thing is when you go into the literature, and then again, there come this kind of what is true and what do you find? If you Google, you find everything. But I mean, Jan is working on a network approach to enrich this more. So there is, well, this is still known. It's part of the U1 spliceosomal system. There's a paper showing that if you knock down U1, you actually affect amyloid precursor, and that, of course, is thought to cause Alzheimer's. So what all this taken together now means for us, because there's this trans association, that there must be something where the ApoE variant, what is the link between the ApoE variant and actually the U1 protein? And it's supported by experimental evidence, because we measure these proteins in this case, 1,000 people. Another thing is cancer immunotherapy. So elotuzumab targets SLAMF7. So this here is like the SLAMF7 concentration as a function of 2 different variants. And you see there's one major signal here that's only a small genetically dependent, but then there's a second, and if both are in the minor allele state, you actually find almost no signal on the SLAMF7, which would then mean that these people probably don't have SLAMF7, and then giving them something that targets at SLAMF7 certainly doesn't make sense, and it replicates in another cohort as well. Again, the caveat here, we try to follow up on this. It's much more tricky, so it's not like that, but it starts to giving you a point where you can try to understand what it means, and especially here to say this is a protein target that you could use for drug response. All right, and now just to wrap things up, what we do in Qatar. So you have the link now between, if you want to bring everything together. So in Qatar we have the advantage, or now they get a bit more tight on the budget, but the advantage to really be able to fund all kind of measurements. So we have a small cohort, through all tools we have on them, so all the platforms, we did genetics, methylation, RNA-seq, circulating microRNAs, proteomics, and glycomics on them. And I think what I should mention, we also have now Qatar Biobank, so we go to a bigger study. So I think they have now 20,000 people, 3,000 have already somologic data from Cornell. And now the next steps from here is, so we're now trying in Qatar to find a way, how can we bring that to the clinic? And one idea, do you know this chart, the biopassport? So in Qatar, there's also a very strong interest. I mean, the football world championship will be there, and they're extremely interested in sports there. There is the anti-doping lab, one of the few accredited ones from WADA. So this here is from WADA, the athlete passport. And the idea, I wrote metabolite here, and in reality it's erythrocytes. But the same idea can be used for metabolites. The thing is, you have an athlete, he has a certain value here on whatever parameter you have. In the population, you have a huge allowed variability. But then if you measure people more and more often, you see that many parameters stay in a certain range. So the allowed range, the personal normal distribution shrinks. And then if that person jumps out, he's flagged in the passport for doping. So we did a study a few years ago where we looked into metabolomics 7 years apart. And what we found is that actually 40% of the people, we could identify them one-to-one by having the strongest correlation between the 7 years. And 95% were actually very strongly correlated. But then there was 5% that changed a lot, and they wouldn't be seen in PCA plots. So that means these people changed over the 7 years. So what we now think we can do is, if we take people in an initial visit, so what you would have here is the normal distribution of any marker, but then the personal variation is actually much smaller. And that's the idea of the passport as well. And that's also the problem why metabolomics biomarker don't work very well, because in the overall spread of many metabolites is very high, so you need a very strong effect, and due to homeostasis, people stay in their range. But if you go and measure them over time, you can establish that range, as in the passport, and then if they jump out, then you know there's something wrong with that person. And that would be a way of personalized medicine using metabolomics on a general way, not a cancer way, a general way. I mean, cancer might also be spotted by that. And then once you have one marker, you would have, of course, many, and then you go to all these different studies we've done in the meantime between all the population studies. So we have information on diabetes. We did something, of course, on cancer, lifestyle factors, all kind of things. I don't have to go into the details here. And then you could come up with scores that needs to be learned, could be learned on things like the All of Us studies, all kind of things there, and then come up with scores. And then try to say if over time these scores vary, I could use them for different applications. And again, for the Qatar thing, the first thing where we could probably go, the question is now, how do we move this to the clinic without getting into trouble with FDA and something like that? So one first thing could be direct to customer things for fitness, where you could say, where's the post-person optimum, things like that. We also have a project now with horses, so they have horse racing in Qatar. So can we predict whether a horse will finish a long distance race? And then through this mean, slowly go into clinical application, where you could actually also predict something on individuals. Right, and that's something we just started in Qatar. We try to create a company for that, where the idea would now be to bring together the metabolomics, all the different local partners. Here the central part is something we do with Jan, where we have all the different computational systems, biology things, networks, and so on. And then in the end products would be different kinds of readouts, scores, reports, and I'm thinking very much about your ATAC thing for the cancer, that we could have other reports here that we could sell initially for the sports, maybe for the racing, and then if we get FDA approval, we could also do them for clinical application. Right, and with that, I would like to thank you very much. So this has been funded by Qatar Foundation. There's a lot of people involved, so I can go through all of them. What I would like to stress, we're really working like an international lab, so I'm still working very much with my former group in Munich, and of course, Jan, who is now here, he comes from Munich, and also many collaborators in London. And with that, thank you very much. And I hope you answer questions. So, hope I'm not too much over time.
Time for one question.
Yeah.
In terms of your in silico, looking at the metabolic pathway, say cancer,
Yeah.
based on metabolomics of cancer cells, do in silico inhibition, let's say, well, you inhibit this.
Yeah. Well, there are already very good examples published very highly, not from ours, but where you have like, if a cancer has two genes on a pathway, and if one is knocked out by a mutation, then the other one becomes essential for the cancer. And if you identify that using TCGA data and stuff like that, then you know if I knock out the other one, only the cancer will be affected. And that's what they call the co-lethality. And I think these are the low-hanging fruit. That works already. But the idea is to make that a bit more complex to identify maybe not so evident links on pathways that could be targeted.
Do you do that in silico now?
Yes. No, we've done already something on glutaminase inhibition. So there was a study in glutaminase inhibition. The cells would actually not die, but when you do metabolomics, you can analyze their pathways. And what Anya found is that if you use a malaria drug, you could actually inhibit the rescue pathway that these cells actually generated. So it's not only in silico. What you need is the combination between in silico and metabolomics data. So what's important is also to have the ability to get cell culture organoids or even tumor samples and get their metabolomics profile to bring that together.
His question was, you're talking about lethality
Yeah. Yeah, that's the idea. The synthetic lethality is something that's established, but there's only a very few examples like that. Now, if you have like the whole pathways, you may find synthetic lethalities that are not that evident. And that's a bit the idea here, to bring together all this knowledge. And that's something we are not there yet, but that's something we work on and try to find ways of identifying that with systems biology tools.
You can predict which horses are going to win the race.
Well, the aim is not actually which horse wins. Actually, it's a long distance runs, 100 kilometers. They drop out, and it's very embarrassing and also bad for the animals. So the idea is rather to predict which ones would not finish the race so that it doesn't start. It's not about winning. I mean, there you do doping. You don't talk about you do doping. Nobody. But it's more about making sure that they don't. And the idea is really more like an opportunity to develop these tools. Like for the sports, we have like Bayern Munich comes in winter to Doha to train. So if we could get like a football club training over 4 weeks and get blood all the time from all the players, I mean, you could make enormous interesting studies with them. And that's a bit what we're aiming at. But well, it's there's IRB, there's policy, there's a lot of kind of stuff to overcome.
All right.
Right, thank you.
Thank you, Carson, for a very exciting talk, and definitely some good opportunities to connect with him on several topics. Our next speaker is Professor Ivan Devlamic. He's an assistant professor in biomedical engineering at Cornell, Ithaca, and he will talk about the liquid biopsy for infectious diseases.
Good morning. So the goal of my lab is to develop genomic-based medicine technologies that can be used to study and diagnose infectious and immune-related disease in humans. And what I'd like to do today in this talk is I will try to convince you that DNA sequencing-based assays, particularly those that are targeting molecules of circulating cell-free DNA, small pieces of DNA that can be isolated from non-invasive samples such as urine and plasma, that those assays offer a lot of potential for infectious disease diagnosis. There are many outstanding challenges for the diagnosis of infectious disease. First of all, there's more than 1,000 microorganisms that cause disease in humans, and conventional diagnostics such as bacterial culture only detect a very narrow range of these pathogens. Second, often invasive biopsies of infected tissues are required to make a diagnosis. And then third, and this is a point that is less appreciated, there's a difference between infection and infectious disease. Finding a microorganism in a clinical sample or finding the DNA of a microorganism in a clinical sample is often not sufficient to decide that a patient is sick because of the presence of this microorganism. And so what I will try to show you is that cell-free DNA can offer answers to these remaining challenges. So cell-free DNA is just simply DNA that floats around in the bloodstream or in urine. This DNA is fragmented. It comes from cells that die. Whenever cells die, their genomes don't disappear magically, and bits and pieces can end up in the bloodstream. Cell-free DNA, in particular in combination with DNA sequencing, offers tremendous applications for diagnostic medicine. For example, non-invasive prenatal testing, which is based on the fact that there's some fetal DNA floating around in the maternal bloodstream. Cancer screening based on the fact that you can find tumor DNA in the bloodstream of cancer patients. Transplant monitoring, something that we've worked on quite a bit, and I'll show you just a brief bit of results there. But what I would really like to focus on today is the potential to use circulating cell-free DNA as an analyte for infectious disease. And so the first question we need to address is whether there is indeed microbial cell-free DNA present in plasma. And the answer is yes. This is something that we discovered when we analyzed plasma cell-free DNA sequencing data sets that we acquired for transplant patients. We had acquired this data for a different purpose. And when we were studying these data, we found that some sequences that were comprising these data sets didn't quite match the host genome. And this bothered us. We digged a little deeper, and we found that some of these sequences actually were a perfect match with the genomes of viruses and bacteria. And that allowed us to sort of recapitulate the virome or the microbiome that exists in the plasma of solid organ transplant recipients after solid organ transplantation. We studied the time dynamics of the viral component of the microbiome, the virome, and what I'm showing here is the composition of the virome early after transplantation. This is when the patients still have a functioning immune system. You need a functioning immune system to survive this the surgery, and this is sort of the average composition one sees. The viruses I would like to focus you on are colored in orange. They're anelloviruses. These are single-stranded DNA viruses that are extremely ubiquitous in the human population. It's very likely that everybody in this room is infected with those viruses right now. And this is what happens with the virome when the patients become immunosuppressed. You can see that these anelloviruses start to take over and become much more dominant. When the drug doses are tapered off because the risk of rejection is reduced, you can see that the virome switches back to its original state. And so we argued in this work that you could use the titer of these viruses as a way to gauge the immunocompetence of an individual, which is something that is now being done at European hospitals via PCR assays. But it doesn't really address the question whether you could use these molecules to do infectious disease diagnosis. And this is something we've, in my group, have tried to evaluate in the context of urinary tract infection. Why UTIs? It's one of the most frequent medical problems that patients present with in medical offices. It's the most common infectious complication in humans. In fact, 30 to 40% of specimens that are received by microbiology lab, like the one that Lars Westblad runs here, are urine specimens. And they're sent there for bacterial culture. To screen for infection. And bacterial culture, unfortunately, it's a great technology. It's been around for more than a century. It's very cheap. It's easy to set up, easy to interpret, but unfortunately is only responsive to those organisms that like to grow in culture. And so it's very prone to false negatives. It also tends to be quite slow, as I will show later. And so what we wanted to ask is whether cell-free DNA is a useful analyte for UTI. And to address this question, we teamed up with a group of kidney transplant physicians and researchers here at Weill Cornell Medicine, including Manikam Sitantiran, Darshana Dadania, John Lee, and Lars Westblad. And we wanted to specifically evaluate this in the context of kidney transplantation because UTIs are extremely prevalent in kidney transplantation. About half of these patients will develop a serious UTI in the first year after transplantation. They will also often deal with viral disease, including BK polyomavirus disease, which can cause serious injury to the kidney. And so we collected samples from these individuals, and I'll just briefly mention that we used a molecular assay that was developed by my student in the lab, Phil Burnham, one of the first few students to join the lab, a single-stranded library preparation to prepare libraries for sequencing for these molecules that turns out to be much more responsive to very short pieces of DNA because it doesn't require size selection as double-stranded ligation assays use. And so you can see that here in the length profiles measured for mitochondrial cell-free DNA and chromosomal-derived cell-free DNA. We've measured the lengths here by sequencing. By doing paired-end sequencing, you can get a little bit of sequence information from both ends of the molecules. You can map these back to the genome and then ask what is the distance between the leftmost and rightmost coordinates of these two sequences. And so that gives you the length of these molecules with single base pair resolution. And you can see that we're recovering much shorter pieces of DNA. We applied this to urine. We found that urinary cell-free DNA is much shorter than plasma cell-free DNA, and that we were able to make robust DNA sequencing libraries from just 1 milliliter of urine. You'll see that if you look at these length profiles, you see this wiggly pattern. This is not noise. It's in fact due to the fact that the cell-free DNA is associated with histones. It's protected from degradation by nucleases by being bound to histones. And DNA makes contacts with histones every 10.4 base pairs, which is the helicity of double-stranded DNA. And you can see this interesting piece of biology reflected in these fragment length profiles. You see that also in the fragment length profiles of certain viruses, including Polyomavirus, which is known to hijack histones in the nucleus of the host and therefore also has this wiggly pattern. Other viruses don't do that. Just an interesting side note. But of course, we wanted to ask, can we use this to screen for infection? And the first thing we looked at, together with Darshana Dadania, was BK virus disease. And we asked, is there more polyomavirus disease in the urine of these patients when they have this disease versus when they don't have the disease? Diagnosis here in the clinic is done by biopsy. We find that there's lots more BK polyomavirus disease in the urine of these patients. In fact, in some patients, half of the molecules of DNA is derived from this virus, which is quite remarkable given the fact that the viral genome is about 1 million-fold smaller than the host genome. So that means that the genome copy numbers are very, very high. Of course, because we're using a DNA sequencing assay, we can look at any virus, and what I'm showing here are all the viruses detected in this cohort, in all the samples where we detect at least one virus. And the red crosses indicate positive clinical tests, right? And so the dots are the results from our assay. And what you see is that all the red crosses are associated with a dot, but you'll also notice that not all the dots are associated with a red cross. And that's not because the doctors didn't detect this particular organism in the urine. It's simply because they didn't do the test, okay? And so that just shows that current clinical protocols that rely on assays that ask one specific question often miss out on a lot of the biology that happens here. Of course, you can ask, and Lars will ask, do we care, right? Do you care about the presence of these viruses? Are they causing disease? And that's something that we'll address in the later part in the talk. We also investigated the correspondence to bacterial culture. And we found, by comparing our results against urine culture, that the agreement was great. There was just one sample where we had a discordant readout. We went back to the clinical records for that individual, and you will not be surprised to hear that we concluded that we were right and the clinical testing was wrong. I showed this work from Stanford. There's a startup that came out of this work, and they've developed a metagenomic cell-free DNA sequencing test. It has the following workflow. A blood sample is collected, and it's then shipped to California to a centralized facility where a sequencing assay is performed, and then the results are reported back to customers. This particular test has been commercially available for over a year now, and it's fun to see first results from customers come out. And so these are studies that the company was not involved in that report sort of on the utility of this particular test. And so this is a nice result from a pediatric hospital in Chicago where they've done 100 Carious tests and compared to standard microbiological testing, including several testings done on invasive biopsies. 54 invasive biopsies were performed on 42 patients. And the results are quite interesting, actually. So first of all, only about half of the biopsies yielded a diagnosis. So these are the results from the standard testing. And so 54 biopsies were performed, only about half of those yielded a result. Nonetheless, and most of these biopsies actually could have been avoided. 85% of these invasive procedures could have been avoided by using the Carious test. And in fact, which is quite remarkable, the Carious test was actually more sensitive than the biopsy in many cases. So a blood test screening for microbial DNA in the blood was more sensitive than even an NGS test performed on tissue removed from the infected site. And so that's something that was maybe not expected beforehand, but now there's more studies that have come out, and sort of the consistent picture that is becoming clear is that this particular blood test, a test that focuses on cell-free DNA in the blood, is more sensitive than traditional microbiological testing in many different settings, including the screening for infection of immunocompromised pediatric patients and sepsis, fever, and neutropenia, and so forth. You may ask, is a sequencing test not too slow? Here are results from Carious that show the comparison of the Carious test against standard blood cultures, and it just shows that the Carious test is not only more sensitive, the diagnostic yield is greater, but also faster, even though the sample has to be sent across the country in many cases to the centralized facility in California. But still, this one problem remains, right? The presence of a pathogen is not synonymous with disease. It's not because you identify a pathogen in a clinical sample that the patient is also sick with disease. There's commensalism, there's colonization, and so on. And so in order to have disease, you need to have some type of interaction between the microorganism and the host. And the relevant outcome of this microorganism-host interaction that you want to measure is injury to tissues. And so then that raises the questions, can we quantify injury via cell-free DNA? And this is also something that we addressed before in our transplant study. Here we had used donor-specific cell-free DNA in the blood of transplant patients as a marker of injury. So more injury, often due to rejection, leads to more cell death in that organ, and therefore the release of more DNA from that organ into the bloodstream. Now, in transplant patients, of course, you can easily discriminate DNA that is released by the transplant organ from the DNA that is released by other cells, simply because the donor and the recipient are two different people and they have a different genome. So you can use genetic differences that exist in the DNA from the transplant organ versus the DNA in all other cells. And so we've done this in the context of heart transplants and lung transplants, and we're able to show a while ago that this is a productive measurement, that you can quantify injury in transplantation using that metric, and actually various commercial tests are also available and are being used in clinics today. And so that shows that you can measure injury via cell-free DNA, but it's not a generalizable approach because it takes advantage of these genetic differences. And so to make it generalizable, we decided to focus on epigenetic marks, these epigenetic features that discriminate tissues, that define tissues. And what I'm showing here is just public data, whole genome bisulfite sequencing data acquired by several public consortia and publicly available for different tissues and cell types. And what we do here is just show all of this data on just two plots. We're using dimensional reduction. On the left, we're using principal component analysis, a technique that we all know and love. And on the right, we're using UMAP, a technique that we don't really know but love even more because it separates out these cell types with greater resolution. And so what we did then was to take urine cell-free DNA and apply bisulfite sequencing to those samples to measure the methalome of the cell-free DNA, which is presumably composed of cell-free DNA from different tissues. Okay? And so when we superimpose these methylomes on top of these two-dimensional maps, you can see that the methylomes span a wide range on these maps, which indicates that the tissue composition of these samples, the tissues of origin of these samples, is quite different, right? It varies from one sample to the next. And you can then use simple algebra by taking the methylome of the mixture and all the references to tease out the tissue sources of the cell-free DNA. So now we can measure the tissue contributions, the contributions of different tissues to the mixture of cell-free DNA that exists in the urine. And to verify that this approach is valid, we compare here the fraction of kidney-specific DNA in the urine against donor-specific cell-free DNA. We had performed this measurement again for kidney transplant patients, and we had this nice orthogonal measurement available. We find a good correlation, and we're satisfied. Okay, so we can quantify kidney DNA, but you can also quantify DNA from all possible other sources in the urinary tract. And then you can start measuring, you can do this in relative terms, but you can also measure the absolute amount of cell-free DNA from different tissues. On the left, we do this for patients that have no UTI or no viral infection. You see a lot of dynamics, lots of injury early on. Then that injury subsides and the patients go through a period of quiescence, which is very much in line with what clinicians see in their daily practice. But when we focus just on a few examples, for example, the case of patients with BK polyomavirus nephropathy, this disease due to the virus that replicates in the kidney and therefore causes injury, you can see that we see lots more kidney-specific DNA in the urine for these patients and for patients that are normal. So indicating that you could use this type of approach to measure injury due to infection. There's some other examples. In the case of UTI, we find more bladder-specific cell-free DNA in the urine. We also see a nice correlation between kidney DNA in the urine and the amount of creatinine in the serum. Creatinine is a waste product of muscle injury and is removed from the blood by the kidney. And if you have poor kidney function, then often that is associated with high creatinine levels in the serum. And so creatinine is a sort of a surrogate marker of kidney function that is often used in kidney transplantation. And we just showed that there's a relationship between cell-free DNA derived from the kidney in the urine and creatinine in the serum. Then the remaining question is, of course, can you still use this bisulfite sequencing assay as a metagenomic assay? Can you identify different bacteria and viruses using that assay also? That is not immediately obvious because if you do bisulfite conversion, for those that know the assay, you're converting all the cytamines into thymines, the unmethylated cytosines into thymines. And bacterial DNA, viral DNA is largely unmethylated, so you're reducing the alphabet for those molecules from 4 letters to 3 letters, thereby reducing the sequence complexity, and therefore it may be more difficult to map back those sequences to bacterial reference genomes. Nonetheless, we find that after bisulfite conversion, you can still accurately quantify DNA derived from different bacteria and viruses and so on. We see a nice correlation between the metagenomic assay based on bisulfite sequencing and the metagenomic assay based on conventional sequencing. Okay, so that brings me to the summary. I've shown that urinary cell-free DNA is a versatile analyte for monitoring of UTI. And I've shown a cell-free DNA assay that can inform the tissue injury response to infection. We're glad to see that our team has been expanded with Mike Satlin and Ilana Brito and Chris Mason. We've recently put in an R1 that is pending, and that will allow us to explore these concepts in much greater detail. The concepts that I showed at the end could also lead to a much more generalizable measurement of human health. It could lead to a blood test for injury that could be very useful in many disease settings. And we've been pursuing that too, specifically in the context of immune-related disease, but that's a story for another day. I'd just like to acknowledge the group. This work was done by 2 students in the lab, Phil Burnham and Alex Chang, and I am open for questions if there are any.
Fascinating stuff. I'm curious about the applications of heat transplantation. So is it possible to screen from the organ, if you take a white biopsy, for example, from the transplanted organ, is it possible to screen for the, the whole virome in that sample in order to predict an organ-specific virus sequence that you would look for?
It would be.
When you repeat transplant, you would be able to pick up the next one.
Yeah, such thing would be possible. A question that often comes up in transplantation, in particular, for example, in the context of BKVN, I think is whether that is donor derived, whether the virus was donor inherited or recipient derived. And what you suggest, the type of assay you suggest, would allow you to address such questions. So I think it would be interesting indeed. Yeah. No, I don't think so.
All right. Thanks, Evan, for a great talk. Our next speaker is Professor Paul Soloway. He's a professor of molecular genetics at Cornell Ithaca and in the Veterinary College of Medicine. And he will be speaking about single-cell characterization of a neurodevelopment disorder. Thanks.
Well, thanks to the organizers for the symposium, and I also want to thank the folks that were presenting posters last night. I really enjoyed them quite a bit. Some of this work will be quite basic. It is intended to discover some mechanisms underlying disease states, and I'll talk about some data that we have for neurological developmental disorders, and I'll talk about also some initial data that we have for metabolic disorders. So the work that I'll talk about with Down syndrome involves a graduate student, Roman Spector, who recently graduated from my lab. And I have a collaboration with Bethany Cummings' lab at Cornell. A student in my lab, Blaine Harlan, has been doing some work with a mouse model for bariatric surgery. And if I have time at the end, I'll also describe some methods for optimizing the workflow for a single-cell ATAC-seq. And I'm also going to make a bit of a shameless plug here for the single-cell club that we have ongoing between the campuses. This has been something that has been spearheaded with Ben Cosgrove, who's here, Jen Grenier, and myself. If you want to join a listserv, feel free to add the subject line of Join Chromatin. We have meetings on both campuses, we have talks on both campuses, and we Zoom these meetings back and forth. So these are very much work-in-progress meetings on a monthly basis. So I get interested in this mouse model for Down syndrome. Within this mouse model, about 132 of the 225 coding genes on human chromosome 21 are triplicated. But with this mouse model, there are a lot of the same craniofacial defects that are seen in human, and a lot of the cognitive deficits that are seen in human are also recapitulated in the mouse model by a variety of different tests. And the reason that I get interested in this model is because of work that's been done by collaborator Barbara Strupp at Cornell. who in some of her breeding of these animals took females that had the TS65DN mutation, and prior to their pregnancy and throughout lactation, she fed them a choline supplemented diet, and the control animals just received the standard control diet. And one thing that she found in her testing, her expertise is as a behavioral psychologist, is that the TS65DN animals that were fed the control diet had more error rates in a spatial memory test, and these deficits are normalized by the maternal choline supplementation. And again, this is a fairly complex trait relying on spatial memory. Looking at another task, focusing on attention, the same kind of deficits are seen in the TS65DN mice born to control-fed mothers. However, the maternal choline supplementation of those mothers normalized a lot of those attention deficits. So these are two very complex traits that have a variety of different etiologies and underlying mechanisms. And the fact that these are suppressed by a fairly simple nutritional intervention is very appealing to me, and it provides some further opportunity for discovery that I'll talk about. But in trying to understand the basis of these deficits or how these deficits are normalized, we applied ATAC-seq to the materials from the brains. And for those of you who may not be familiar with it, ATAC-seq begins with nuclei with chromatin that has nucleosomes on it. And the TN5 transposase is able to integrate oligonucleotides of your choosing into the genome at open chromatin sites. Open chromatin sites tend to be those that are gene regulatory in nature in specific cell types. And by applying these oligonucleotides that are then compatible with PCR of your choosing and Illumina sequencing, we can essentially map where open chromatin is in the genome. And this is a method that was developed by Jason Buenrostro when he was in Will Greenleaf's lab at Stanford. This has been modified more recently into a single cell profiling approach. And in this particular approach, we begin with the oligonucleotides. 20 of them can be arrayed on a 96-well plate to give 96 independent barcode combinations. And then by putting 2,000 to 4,000 nuclei in each well, we can then perform this tagmentation or the integration of these oligonucleotides. And again, these will mark sites of open chromatin. We can then pool all these from an individual plate and then rearray it onto another plate that has yet another independent 20 different oligonucleotides that together form 96 different barcode combinations. And in the PCR assay, we can then amplify the materials. And if we first sort 25 nuclei into these plates, we can have a very high likelihood that we're looking at single cells, and we've done the control experiments that validate that that when we do have two cells with the same 4 barcode combinations, that's in the single-digit percentage rate. So here are some of the data that we get back. These are just describing the various samples that we have. We have duplicates of the TS65 DN animals, and we have biological duplicates and technical duplicates as well for each of these. And what you're looking at here are the read outputs per sample. And these are, each dot represents an independent cell type. So here are the number of reads at the top that we have per cell, about 4,000 to 5,000 reads per cell. And here are the number of cells that pass the quality control metrics that we impose, which is a minimum number of reads per cell. And these are the data that we then apply to analysis. Now, the reason that we want to go to a single cell format for this is because the brain is obviously a very complex structure. Of course, we know that there are neurons of a couple different varieties. We know that there are a variety of different glial cells. But these represent unknown populations to us. The population substructure of these cells is likely to be much higher than we appreciate. Furthermore, if we applied a biased approach, namely to sort individual cells out, we may erroneously underestimate the complexity of the cell types there, because you can have an antibody that might identify neurons. But again, the complexity of the neurons that we have is something that is perhaps underappreciated. So by applying a single cell format, we can be completely unbiased in our analyses of the cells that we get back. Now, Ewan described this UMAP tool that was developed by Cole Trapnell's lab, and we applied this as well to discern the heterogeneity of cell types within the cortex of mice. I did not mention this, but we used the cortex. And these are the clusterings that we get back in this 3D UMAP plot. We identify 12 different cells that we identify as excitatory neurons, and I'll describe to you how we make that identification. 8 different cell types that identify as interneurons, 3 different oligodendrocytes, 1 astrocyte population, 1 endothelial cell population, and 1 microglial population. And this might actually even still be a bit of an underestimate because we characterized only 11,000 independent cells here, and if we increase the number, we may have the capacity to further refine these cell assignments. And additionally, if we consider other domains of the brains, we may have even further complexity discerned. So how do we begin to identify these cell types? These are just browser shots of these different clusters. We're showing 26 different clusters over here. We're showing browser shots for 6 different genes here that are canonically identified or active in each of these different cell types. And you can see, for example, that these cell types that we're identifying as excitatory neurons are enriched for reads in the NeuroD6 gene. So this is an unsatisfactory way to identify individual cell types. However, it's illustrative of the kind of approach that we can take that's applied in the next slide. So here we have, again, a variety of different genes that are canonically identifying individual cell types. And the circles here, coded by color and size, report the enrichment of reads at open chromatin within each of those genes according to each of these 26 different cell types that we identify over here. So for example, we can identify somatostatin positive interneurons because somatostatin is an enriched open chromatin site within cluster 19. We can also identify parvalbumin interneurons, VIP positive interneurons, and there are other interneurons that are not falling into any of these classes. We can identify excitatory neurons that map to certain layers within the cortex, and these are identified by other analyses that have been histologically. And we can identify oligodendrocyte precursors, for example, among the pool of cells that are, again, glial in nature. Now, one of the nice things about this UMAP strategy that differs from other comparable tools like t-SNE analysis is that clusters that are closely mapping to one another in this 3D space are frequently related. And this was the original UMAP plot that I had shown you before here on the left. And on the right, these have been recoded according to the individual cell types that we've identified. So the fact that the excitatory neurons are co-clustering in UMAP space provides a sanity check for the accurate identification of these cell types. The next 5 slides or so just, again, provide another sanity check for our identification of these cell types. And these take advantage of gene ontology categories. Again, we show a variety of different categories here that I'll scan through according to each of these 6 broad classes of cell types that we've identified. And reassuringly, within both neuron classes, excitatory and interneurons, we have GO categories that we would expect to find in these cell types, focusing on, again, membrane potential and synaptic transmission. Within the oligodendrocytes, we expect to find gene ontology categories that are relevant for gliogenesis and exon achievement, and that's indeed what we find. Within astrocytes, we expect to find markers of metabolic processes, and we find those. Within microglia, we expect to find gene ontology categories that correspond to immune responses. We find those as well. And within the endothelial cells, we do expect to find GO categories that are relevant for angiogenesis, and we find those too. So these provide, again, a sanity check that our identifications are accurate. So what we really want to do is to compare the TS65 Down syndrome model with their wild-type counterparts. So they're coded over here. The 2N refers to the wild-type animals, the TS65DN here in green. And I'm showing here just these broad categories of cell types. And we can see a 50% increase in interneurons within the Down syndrome animals. That's one of the statistically significant correlations we identify. And also there is an underrepresentation of endothelial cells. If we break these categories down into each of the individual cell clusters, we can find that the most abundant of the cell types in our data set, namely this category 1 of excitatory neurons, are also reduced significantly significantly in the Down syndrome animals, even though as an overall group, excitatory neurons themselves are not underrepresented. But again, if you focus on an individual cluster, an individual category of excitatory neurons, we can see a significant deficit. Again, with the interneurons, there are two main classes that tend to be overrepresented, that are driving the elevated levels of interneurons in these TS65D mice. And again, here are the endothelial cells that seem to be underrepresented. Now, we can also take a look at gene ontology and disease ontology categories, specifically at differentially accessible peaks between the Down syndrome cell clusters and the corresponding wild-type cell clusters. And one thing that I'll point out over here is that those endothelial cells that are underrepresented seem to be enriched for markers that relate to cell cycle control. So one of the things that we speculate is that in the TS65DN animals, the lower abundance of endothelial cells might be due to their overall lower proliferation, and this may be part of the underlying pathology of Down syndrome. So beyond the cell categorization, we can do another level of analysis. We can identify transcription factor motifs that are enriched in each of these broad cell categories. And I'm not going to go through all 26 categories, but I'll give you illustrations of these major categories, the excitatory interneurons, et cetera. And we can find specific motifs that are enriched at the open chromatin that we identify. And again, this provides another sanity check for us. Neuro D1 and G2, for example, are known to be involved in specification of neurons, and motifs for these transcription factors are overrepresented in the excitatory neurons. LHX is a marker for, or is a transcription factor involved in interneuron development, and again, that is enriched in the categories of cells we identify as interneurons. Again, we can go through a lot of these different cell types and all the 26 different cell categories, but I'm just giving you an example of the kind of outputs that we receive. We can also take a look at transcriptional motifs at peaks that are changed within these cell types between Down syndrome animals and the wild-type controls. Again, LHX, which is the marker or a transcription factor important for interneuron development, is significantly upregulated in the interneurons of the Down syndrome animals relative to the wild-type animals. And again, this is probably part of the underlying etiology of Down syndrome. Additionally, within the triplicated domain of Down syndrome, there are a number of different transcription factors encoded, and these transcription factor motifs are also enriched at the targets that are differentially accessible between Down syndrome and the wild-type animals. And again, the potential for overexpression of these transcription factors within these triplicated domains is potentially related to the underlying pathologies associated with Down syndrome. So with these baseline data in place, I'm very eager to go back to our freezer. We have brains from all of these animals that have been subjected to all these behavioral testing. And again, they provide a wonderful resource for basically a chemical suppression experiment. So if we repeat those kinds of analyses on the brains from these animals, we will presumably identify some cell types that are normalized, some particular genes that have normalized open chromatin sites, and some transcription factor motifs that may be enriched at those sites. And those provide a filtration mechanism for us to further narrow down our focus on which genes, which cell types, and which transcription factor motifs are most underlying, again, these complex traits, two examples of which I show here, spatial memory and attention. So in the remaining few minutes, I want to turn attention to a data set that's very much emerging right now. This arises from an interest that my colleague at Cornell, Ithaca in the vet school, Bethany Cummings have. She's applied a mouse model for vertical sleeve gastrectomy, which is frequently used in humans as a bariatric surgical intervention for obesity. And this has the capacity to normalize a lot of the metabolic deficits associated with obesity and diabetes. And interestingly, people respond very quickly to bariatric surgery even before they experience any weight loss. So the normalization of metabolic deficits that are seen in obese patients, again, can be normalized by this process even before they lose any weight. So we got very interested in the potential role of adipose tissue in this process. And particularly, white adipose tissue undergoes a beiging process in bariatric surgery patients. And in beiging, there is an elevated level of mitochondria that arise. There's an elevated oxidative phosphorylation rate, and there's elevated uncoupling that occurs as well, leading to a higher overall level of energy expenditure. And this normalizes some of the glucose homeostasis. So we've applied some of these same methods to adipose depots isolated from animals that have been subjected to bariatric surgery. So the protocol in Bethany's lab is to take 8-week-old male C57 black 6 mice, feed them a high-fat diet for 8 weeks, and then perform the vertical sleeve gastrectomy or provide animals with sham surgery, and then continue the dietary interventions for another 2.5 months, but in this case also adjusting the food intake of the animals so that they are overall weight matched. And this is sort of an important control, and so we've applied single-cell ATAC-seq to that. Again, just some technical details. We have a higher read count per cell with this data set than we had with the brain, and part of the improvements that we implemented was to perform tagmentation at 50 degrees, so those of you who are doing this might want to consider that. Here are the number of cells that we have that, again, pass these quality control metrics of 1,000 reads per cell. We can do the same kind of clustering as we did before, and we have 16 clusters. Again, this is a simpler tissue type than the cortex of mice. And I'll just illustrate what we have found among these few different clusters. Our largest cluster is cluster 1, identifies one of 3 different types of adipocytes emerged from our analysis. And one of these adipocyte populations is increasing in abundance in the vertical sleeve gastrectomy animals. And again, we're identifying these as likely adipocytes based on some of these gene ontology category markers that we expect to see in adipocytes. We see a fairly high population or heterogeneity of cells that we identify as neural in origin. And again, these have markers that we expect to see in neurons. And interestingly, we have one class, it seems the most abundant class of neurons, seems to be present in reduced abundance in the vertical sleeve gastrectomy animals. We can also find a couple different categories of immune cells, one of which is elevated in abundance in the vertical sleeve gastrectomy. And again, these have gene ontology categories that help us identify these as immune cells. We can identify some other cell types that we're identifying as likely to be stromal, simply based on collagen fibril organization, although we have much more data analysis to really solidify these identifications. However, one of these clusters is clearly present in higher abundance in the vertical sleeve gastrectomy animals. And we have other cell types that have no significant changes between the two endothelial cells, and a gonadotropin responsive cell type whose origin we don't know quite yet. But we see this as being a potential discovery tool, and if from this emerges markers for recovery from metabolic disease, or in the case of Down syndrome, perhaps alterations as a result of nutritional interventions that are of benefit for patients, we see these tools as being potentially beneficial. I'm pretty much out of time, so I won't have time to go through this last optimization or improvement in the workflow, but for those of you who might be applying single-cell ATAC-seq, again, to your own favorite cell populations, one thing that we have found with our adipose tissue is that we can freeze nuclei directly after the tagmentation stage. So if you're going through a whole bunch of tissues at once, it's beneficial perhaps to coordinate all that workflow within one day for your nuclei isolation and your tagmentation. You can then freeze them and then occupy the next few days on the FACS core for sorting before doing the PCR and the PCR cleanup. And I'll just show you that when we do this, what we're showing here are frozen tissue versus not frozen tissue. And basically, we see the same cell clusters, and we see no differences between the two workflows. So I'll leave it at there, and I'll be happy to answer any questions you might have.
So a two-part question. Do the clusters that you came up with in the down mouse model correlate with the electrophysiology of different neuronal types? And the ones that are changed in the down model, are they neurons that express cholinesterase transferase or various alpha subunits of the S2?
Okay. So the question gets at the physiology of the cell types that we're identifying in these systems and the differences that might exist between Down syndrome and not. I'm going to have an unsatisfying answer for you, and basically we have no reliable ways to correlate the individual cell types that we find with the electrophysiology that has been done on other cell types, because to get that good concordance is quite difficult. And so there certainly can be. So for example, with the interneurons, those that are parvalbumin positive or VIP positive, these provide some means of correlation. With respect to your second question about acetylcholinesterase or acetylcholine transferase, that's a gene that I did not look at specifically. That would be worth looking at. So I know Ewan has some methods for a higher throughput in situ hybridization than currently exist. But the old-fashioned methods basically did one probe at a time, and you had as many potential fluorophores as you could manage on those probes. And again, the throughput's limited. I think even with some of the newer systems that Ewan's lab has been developing, they're still in the single digits of thousands. It would be wonderful to basically apply some spatial transcriptomics approaches that have high resolution and that have no bias whatsoever. We've not implemented those yet.
All right. Let's thank Paul for a great talk.
Our next speaker is Dr. Yariv Huvris, if I pronounce it right. He's an associate professor and a physician scientist who works at the interface of cancer genetics as well as aspects of oncology. He'll be discussing building patient-specific genetic models of cancer in zebrafish. Thank you.
Thank you very much. It's a real pleasure to be here this morning. Thanks to the organizers for inviting me. So I'm a medical oncologist. I care for patients with advanced thyroid cancer, and in my lab we use zebrafish to model both melanoma and thyroid cancer. And I think it's useful. Some people have never even thought about using fish to model cancer, to think about the historical origins of this idea, actually, because it's related to why we all have jobs, or most of us anyway. So we could start with just by asking, is using the fish to study cancer new? It turns out it's not a very new idea. This was actually based on work done upstate in New York by Gaylord and Marsh in the early part of the 20th century. They were studying trout. I'll tell you a little bit more about this in a second. They were very successful in getting science reporters from the New York Times to cover their work. This is in the pre-Gina Colada era. And they captured the attention of Henry Taft, our 27th president, a Republican, who also has the distinction of being the only president who also was a justice on the Supreme Court. And he wrote to Congress, "We have every reason to believe that a close investigation into the subject of cancer in fish may give us light upon this dreadful human scourge." He requested money from Congress for biomedical research. This is the first time that ever happened. This predates the origin of the NIH, and this is the beginning of the way our federal government in the United States started to fund biomedical research to study cancer in fish. And specifically thyroid cancer, although they had it slightly wrong. This was actually turns out to be goiter, because in the fisheries in upstate New York, they did not understand that they had to supplement the water with iodine. Nevertheless, 109 years has gone by. How much progress have we made? All right, so I'll tell you a little bit about the work from my lab and some other work from the zebrafish community. So zebrafish is a small freshwater fish in the pet store. It's kind of like the most boring fish in the pet store. And, but they have a remarkable set of attributes. They're highly fecund. They're easy to grow and maintain. A pair of zebrafish can mate weekly. They can make 300 embryos a week, and development proceeds from the one cell stage to a complete animal with a beating heart, circulating blood, eyes that move, and organs within 24 hours. By 5 days post-fertilization, all organ development is essentially complete, okay? All of this takes place in water in a petri dish right in front of your very eyes. Okay, so even 300 embryos a week is a lot of new life to bring in the world. Even as the father of twins, I can say that. Because zebrafish development happens in a petri dish in front of us, we have access to this animal from the one-cell stage. And that access gives us a lot of capability. We can perform single-cell microinjection. We can put anything into that cell that you can dissolve in liquid. We can add plasmid DNA, build transgenics, and do lots of genome editing, which I'll touch upon at the end. Here's the first, 6 hours of development. That was gastrulation. So if you want to watch this, just call us up, come by, we'll sit you down in front of a microscope and you can watch the miracle of life every single day. We can house these embryos at remarkably high density in kind of like state-of-the-art aquatics habitats. In the basement of this building is the more or less the first or second largest facility in New York City with a capacity of about 5,000 tanks. These tanks are arranged in bookshelves. It's a closed circulating loop of water, so if you want to study the microbiome of fish, come ring us up. This is a completely climate-controlled facility. These fish are all born in the facility. They're hand-fed 3 times a day. They have 5 different diets, and they're computer monitored at a level of granularity that exceeds the average ICU hospitalized patient. Okay, so this is an intense system, and it allows individual scientists, postdoctorals, researchers, graduate students, technicians, to have really, really large numbers of animals in projects. A single tank can house 30 animals. So I would say the average postdoc in my lab has more animals than the largest mouse investigator at this institution. What have we done with this? So we can take these attributes, speed of development, fecundity, forward and reverse genetics, conservation of cells and organs, and we can build models. So about 15 years ago, a model of melanoma was built in Len Zon's laboratory in Boston, and now we have probably about 50 labs in the world that work on the melanoma model. It's the most advanced model in the zebrafish. I'm not going to talk about it today, but we have a whole suite of genetic, chemical, and cell biological tools that have been applied to that model to do both gain and loss of function studies, and that have led to really, I think, important discoveries. You can see here melanomas are rising on the lateral aspect of the animal, and here we've knocked out one of the pigmentation genes with CRISPR, so it kind of looks like the stripes have been erased. And what you see under the stripes, these silver areas, these are iridophores. These make the fish sparkle. This is a pigment cell that fish have that we don't have, but it's interesting. It's related to the melanocyte lineage. Okay, but today we're going to talk about thyroid cancer. This is a disease that I see clinically. This is a prevalent disease. There are about 56,000 cases in 2017, probably about 65,000 this year. There are 1 million or more Americans living with a lifetime diagnosis of this disease, so I'm sure there are several people in the audience today. Thyroid cancer can recur, and it can cause significant morbidity when it does, but it's not generally a very lethal disease. We have in the United States about 2,000 deaths per year. In terms of the genetics, it's very interesting. About 60% of thyroid cancers are characterized by a BRAF mutation, and this is the same mutation that's found in about half of all patients with cutaneous melanoma. About 10 to 30% of thyroid cancer patients harbor RET mutations, and I'll tell you a little bit more about this in a second. And unlike melanoma and other cancers that are exposed to the environment, thyroid cancer is a very mutationally quiet disease. So the average BRAF mutant papillary thyroid cancer only harbors 7 to 10 additional somatic mutations. So this is a patient of mine from the clinic that we've been studying intensively in collaboration with members of the Englander Institute over the past 7 years. She's a 52-year-old female. When I met her, with a 26-year history of thyroid cancer. This was diagnosed when she was a young woman. She had a 2.5 centimeter papillary thyroid cancer that had some areas of poorly differentiation. Over the ensuing 15 years, she was treated multiple times with radioactive iodine, suppressive thyroid hormone. These are typical medical therapies for thyroid cancer. And she was well. She was living her life, working very productively. Unfortunately, in 2013, she had a cough and was diagnosed with recurrent disease. It was resected and found this nodule in the lung to be a recurrence of her thyroid cancer. And then when I met her, she had multiple sites of bony metastatic disease. So she had a large kind of golf ball-sized lesion growing out of her calvarium. She had a very painful lesion in her sternum and multiple rib metastases. So this was clearly now a crisis for her after living with this disease for almost 30 years. And we sought to kind of try and understand what had happened to her over this very long time, to go as deeply as possible into this one case. And so in collaboration with Olivier and Andreas Sponer and others in the Englander Institute, we performed very comprehensive exome sequencing, RNA sequencing, copy number alterations. We got her blocks from 26 years ago and performed fluorescence in situ hybridization. And we identified a rearrangement involving the RET gene, R-E-T, and this partner protein, CCDC6, and we could show that that actually was present in her lung and bone mets and present in her initial tumor sample from 1988. We also identified a TERT promoter variant. We could not perform sequencing on her original sample because the DNA was too degraded, and there was a focal p53 deletion. As she was treated, and I'll tell you a little bit more about her treatment history in a second, we performed analyses of cell-free DNA, and we were able to monitor the allelic fraction of RET through treatment. So I treated her, and after she was diagnosed with this rearrangement involving the RET gene, there was a real challenge to try to identify what therapy would be most beneficial for her. And so there was a decision made to treat her off-label with a RET inhibitor, cabozantinib. And she did respond. She had a radiographic response. I think I have some slides on that in a second. And she had, you can see kind of some evidence of a decrease in her cell-free DNA, but then she unfortunately relapsed. So let's talk about RET gene rearrangements for a second. These are present in somewhere between 10 and 30% of thyroid cancers. RET is also rearranged in about 1% of lung cancers and rarely in a variety of other solid and liquid tumors. RET is mutated in the germline of patients with a hereditary endocrine cancer syndrome called multiple endocrine neoplasia type 2. These patients harbor a spectrum of different cancers involving the thyroid and other endocrine organs. There are two FDA-approved kinase inhibitors that have very potent anti-RET activity. They are cabozantinib and vandetanib. These drugs are approved for the treatment of not this kind of cancer, but related cancers. And there are multiple now highly specific RET inhibitors in clinical development that show exceptional promise. These are made from companies like Loxo and Blueprint. So what is RET in terms of its signaling? RET is a transmembrane receptor tyrosine kinase. Its ligand is actually members of the GFR alpha family bound to GDNF and related ligands like nurturin and percephorin. It has a role in enteric neuron development in the GI system during development and also in a variety of endocrine tissues. It signals to the MAP kinase and PI3 kinase pathways, and the kinase inhibitors that have been developed block its kinase activity. When RET is rearranged in thyroid cancer and in lung cancer, these rearrangements result in the expression of a cytoplasmic chimeric protein that is no longer plasma membrane bound and no longer ligand dependent. So it will signal constitutively. And that's a problem for any of those cells. So when PM271 was initially diagnosed, we were obviously very concerned about her clinically, and we realized what a unique opportunity this was to study this disease. And so we actually did not start with zebrafish. We started with a variety of other conventional methods, 2D culture with growth factors, culture with feeder cells, normoxia, hypoxia, defined media, organoid development, and growth in multiple different immunocompromised mouse xenograft models in collaboration with Giorgio Ingrami. And what I can tell you is that we could get these cells from directly harvested, warm, from the operating room to passage in culture a couple times. We could stain them and show that these were in fact cancer cells. We could sequence and find the molecular alterations, but we could never establish a model from this patient. After 1 or 2 doublings, these cells would die. And none of these other approaches were effective. And unfortunately, this is typical in the world for thyroid cancer. It's very, very difficult to establish cell lines or create models. So I would say we spent a year and some very large, unquantifiable amount of money on this. And then a postdoc from my laboratory came to me and said, hey, what about that zebrafish thing you keep telling us about? And actually, for a variety of reasons, I was very skeptical that this would work in the fish because we had previously built a model of BRAF mutant thyroid cancer. And there were some very significant challenges with that model, which I don't have time to unpack. So I did not think that RET was going to be a great candidate. But the postdoc, this is a word of advice for all postdocs, don't listen to your PIs. The postdoc just went ahead and did it. So he expressed this patient's unique fusion. Now we're expressing the human gene under the control of a zebrafish thyroid promoter. And within weeks, he saw the growth of tissue outpouch from the ventral jaw. He could show that this was green. The animals started dying. When we let them grow up a little further, I think you can appreciate here this mass arriving from the ventral jaw in the zebrafish. When we look at this histopathologically, we see this is thyroid cancer. This has multiple features of a poorly differentiated thyroid cancer that also preserves some well-differentiated areas and follicle formation. It's TG positive, it's PCNA positive. So this was really amazing. This is several years ago now, and we created a stable line, and we've been propagating that line, and I think we'll be able to propagate it hopefully indefinitely. Now, what have we learned from studying this model? So I want to take you through some of the highlights and show you some of the unique capabilities. So before these animals grow up into this sort of adult animal, we can study larval animals, which are much smaller and amenable to a lot of different kinds of cell biological exploration. So in this example, what I'm showing you is now what wild-type thyroid looks here. It's a series of discrete follicles that are organized in the midline, in the ventral jaw, and we can stain them for thyroid hormone, T4 in red, which is present in the lumen of these follicles. And the follicles themselves are green because we're expressing, we're staining for that, an epithelial protein. Here, when we express RET-PTC, you can see the organization of this tissue is completely disrupted. It's no longer discrete follicles. It's just growing really as like a mass. There's some follicular formation. There are many, many more epithelial cells. We can soak these larval animals in BRDU to label cells in S phase of the cell cycle, and we can show, as compared with wild-type animals, which have almost very, very few S phase cells, in the RET-PTC expressing transgenic larvae, we have lots and lots of cells in S phase. We can let the animals grow up a little bit until they have small masses. And this is at about 8 days post-fertilization. And we can perform immunostaining for phospho-ERK and show that these cells that are proliferating are kind of engaging MAP kinase signaling. And their wild-type counterparts are essentially not. We can then take these animals, we can make hundreds or thousands of them, and we can flow sort out the thyrocytes from every other cell in the animal. And we can perform RNA-seq analysis. And the RNA-seq analysis, if we perform GSAA, reveals that the cells are very different from their wild-type counterparts and are activating transcriptional programs of mitosis, cell cycle, and cyclic AMP signaling. Okay, so what about treatment? So we can then start to take these animals, and we've had to essentially build kind of the assays for doing this. So I won't have time to show you everything, but I'll show you a few examples. So in this example, we're essentially soaking larval animals from 4 days to 7 days of life, right after the thyroid is essentially formed, in cabozantinib, the same drug that the patient was treated in. And we're looking at the effect of thyroid size and measuring volume by confocal microscopy. Okay, and we've refined this ability. We can count cells, we can look at thyroid differentiation markers, we can treat the animals, flow the cells out, and look at effects on gene expression. If we let the animals grow up a little bit, then we have enough material to perform biochemistry. So now we basically have, in this example, what we've done is we've treated adult tumor-bearing fish ex vivo. So we harvest the tumor, mince it up in a petri dish, treat it with the drug, and then look at the effects on signaling, and you can see the drug does hit RET, phospho-RET, hits phospho-ERK, hits phospho-AKT. Okay, so the model starts to look like something that might be useful in helping us understand how the drugs work. Well, the patient is also a really important model, right? Individual patients. And so what happened to this patient? So the patient was treated with cabozantinib, and we had to do this before a zebrafish model was built, right? I mean, there's a kind of a urgency in the clinic that is different from the lab. And the patient did well. The patient had improved pain and multiple bony sites, had disease stabilization, and I would say, in terms of like from a medical oncology perspective, her best clinical response by RECIST was stable disease. So there was some benefit, but the magnitude of the benefit was not overwhelming. So this sort of leads to an important question, right? Why aren't RET inhibitors more effective in patients like this? We know this disease is driven by a RET expression, and so maybe the drugs are not potent or specific enough. That could be the case. And the newer generation of drugs are highly specific, low nanomolar inhibitors, so we'll see if they work better. Perhaps the tumors undergo clonal evolution during treatment. Perhaps there's an immune evasion component. And finally, the last thing I want to unpack a little bit is this concept of adaptive or really biochemical resistance to drug treatment, and in this case, treatment with a RET inhibitor. So this fourth point is actually the point that we focused a lot of energy on over the past few years. And I think the cancer biology community is still wrestling with and trying to understand this. I want to tell you what this looks like. So for these studies, now we turn to a human cancer cell line, the TPC1 cells that harbor this, paradoxically, this exact same rearrangement. And what we can show in these cells is that if you treat with the drug for 1 hour, 6 hours, you hit phospho-RET, but by 25 hours, let's see if there's another panel here, the signal is, phospho-ERK signaling is starting to rebound here. So the cells are initially sensitive and then they rebound. And I can show you this again here, that basically treatment at 24 hours or retreatment is associated with a rebound in phospho-ERK signaling. Now, we know that this is happening upstream of MEK because if we treat simultaneously with trametinib, a small molecule MEK inhibitor, we can completely abrogate this rebound. Okay, so the cells harboring this rearrangement are learning very quickly, within a matter of hours, how to bypass the effect of the drug and reactivate MAP kinase signaling. So we started to think, well, why could this be? And many other investigators have started to think about SHP2, which is a dual specificity phosphatase that is important for integrating signaling from MAP kinase and other pathways. So we examined this, and in fact, if we treat the cells with SHP2, SHP99, a small molecule inhibitor of SHP2 from Novartis, we can essentially block this rebound, okay? And you can see here that other drugs like afatinib and erlotinib, partially effective, sort of implicating those receptors in these bypass circuits, but not completely effective, whereas SHP2, completely effective. So that leads to a model, right, where these inhibition of these kinases somehow leads to a rebound that is SHP2 dependent. SHP2 is known to promote GTP-bound state of RAS, and so we believe that actually what might be happening is that cytoplasmic RET may be sequestering SHP2. Treatment with small molecule RET inhibitors may release SHP2 and allow it to activate receptors at the plasma membrane and reactivate MAP kinase signaling. So in fact, we can go back to the zebrafish model now, treat with cabo alone, SHP99 alone, or the combination, and show that the combination is markedly more effective in short-term in vivo treatment if we inhibit SHP2. So this sort of leads to then a paradigm for how we might treat future patients who harbor these RET rearrangements and ideas for thinking about combination therapy. So I have a little bit more to show you, but from this body of work, the RET rearrangements, hopefully I've convinced you, can initiate thyroid cancer, and they may be linked to aggressive clinical phenotypes in some thyroid cancer patients, and we know that's the case in lung cancer. Pharmacologic inhibition of RET leads to responses, but also the development of adaptive resistance, and that resistance is biochemical and happens on the time frame of hours. So these models that we've built allow us to dissect, using chemical and genetic approaches, this resistance and identify new targets. So the idea for us in the lab is to use the fish to build patient-specific models and do two kinds of screens. Do phenotypic screens to identify novel drug targets, and build patient-specific models to test specific drugs and specific drug combinations. Now, one of the amazing breakthroughs that we've been experiencing is the advent of CRISPR-Cas9 and genome editing. And in zebrafish, this really is an unbelievable windfall for us. And I just want to show you one example here. So in this example, basically, we've targeted the TYRP1B gene. This is a gene that's involved in the formation of melanin pigment in melanocytes. And so we basically identified a guide that would create a lesion in the terminal exon. And then we created a template here in silico and then synthesized this to basically fuse GFP in frame with exon 7 of this gene. We created a stable transgenic line, and you can see expression of GFP in melanocytes here. So we literally have the capability now to stitch genetic material into the genome of this species anywhere, okay? This includes fluorophores, affinity tags for biochemical purification, locks and flip cresets for conditional alleles, and probably even building larger scaffolds for more complicated genetic engineering studies. So we're really, really excited about this, particularly in terms of its application to building models of human disease. So I want to acknowledge the people in my lab who did this work. Renuka Raman is leading the efforts on the RET-PTC model. She picked up where Jacques Villefranc left off, and she's had a lot of help from Timothy Ullman. Sarah, Katie, and Raul have been pushing the genome editing project forward. We've had great help from Olivier, Andreas, Rohan, and Juan Miguel Mosquera in the Englander Institute, and the PM271 project is a collaborative effort that also involves Tom Fahey and Teresa Scognamiglio. Thank you very much.
So, in the interest of time, I think you can catch up in the coffee break. During the coffee break, we'll move on to our next speaker. The next speaker is Dr. Monica Guzman at Cornell Medicine, and she will talk about targeting the epichaperome as a tailored therapeutic approach.
Good morning, and thank you for the opportunity to discuss this project that is undergoing in the lab. To get started, you might wonder what is the epichaperome and what is the basis of us trying to do this approach. So we really think in the case of our lab, which is acute myeloid leukemia, that is a blood cancer, but this is a concept that applies across all tumor types. In order to be able to tailor therapies, we really think we need to understand the dependencies of tumor cells. And if we understand them, it's going to be easier for us to attack them. The best analogy I can use is if I were to ask you, how do you feel when you misplace your smartphone? And I thought this cartoon was very telling when you see the sun mark, the suntan mark on these people of being just all day attached to their cell phones. So when we ask this question, in order to understand the epichaperome, is if you feel really upset when you lose your cell phone, that means that probably you have a huge dependency on that cell phone. If you feel a little bit worried, maybe your dependency is not as strict. You might be a little bit stressed, or maybe you are actually relieved. So the reason that this happens is, if we put it in this analogy context, is if you are extremely attached to your cell phone, and you feel like you are not going to survive without it, is because that cell phone has a lot of networks on it. So your ability to contact or interact with other people in other ways to network is dependent on that cell phone. For example, nowadays, most of us don't even remember a cell phone number or how to get to a place without asking Google Maps, how do I find the house of my friend or even your own house. So that happens if your cell phone is lost. You're not probably going to be able to find your friend, find their house, or how to get to your favorite restaurant. So, but in other hand, if you are relieved, it's probably because you use that cell phone as a cell phone, meaning you have few contacts, you use it to call some people, and that's about it. But if you need to find people on network, you're able to do it because you remember where they live, you remember their number, and you have alternative ways to network and identify or find those people. So the epichaperome is something very similar. So in the epichaperome context, the epichaperome are machineries of chaperones, which are protein networks that are centered on HSP90 and HSP70. And what happens in this context is that that you have very tightly and highly connected networks. So if you take out HSP90, for example, of this network, the whole network collapses and the cell, in the case of the tumor cells, will die because everything is so interconnected. In contrast, when in other tumors that have low levels of this epichaperome or don't have this epichaperome, or in normal cells, these networks are much smaller. They just, they are still of proteins connected, but they are tinier, meaning if you get rid of HSP90, the cell will have alternative ways to upregulate other compensatory pathways and lead to cell survival. Another analogy I like to use for this is if we were to think about those Jenga towers. So the bigger the tower is, it's probably and the more unstable, it's easier if you pull one block out, the whole tower will fall down and collapse. But if that tower is much smaller and you pull out a block, the tower will still remain intact. So it's something similar. It's a similar principle that we want to establish in the lab when we're starting to focus on the epichaperome and how these epichaperome networks work in cancer cells. So specifically, we were studying the epichaperomes for AML, but we have defined that this concept is true across every tumor type that we have evaluated. It is important to mention that these characteristics of the epichaperome are completely independent of mutations or on any other genetic aberration or cell type that you might think that could correlate to. So what we have found is that there's two types of cells, one that are epichaperome abundant, and those cells that are low epichaperome or epichaperome negative. So what we did in collaboration with Gabriela Chiosis at Memorial Sloan Kettering was to develop tools that would allow us to identify cells that have these high levels of epichaperome or they were epichaperome abundant at the single cell level. So this was done by flow cytometry. What we did is develop a fluorescent probe of a drug called PUH71, which is an epichaperome inhibitor. So this drug is linked to FITC, and we're able to incubate it with cells for a couple of hours, and this drug will incorporate into the epichaperomes. That way, when we do flow cytometry, we can see which cells are abundant in the epichaperome, and those cells will be predicted to be sensitive to PUH71. And also, when we have a cell that has just regular chaperomes, when we add the PU-FITC probe, this probe won't be able to bind, and you will detect it by flow cytometry as FITC negative, and therefore epichaperome low sample. Another thing I want to mention is that we also found that epichaperome abundant cells have hyperactivated signaling networks, which results in pretty much every signaling pathway to be turned on, and that will be relevant in the case I'm going to mention shortly. So what we also know is that these epichaperome abundant cells, when treated with PUH71, they are going to they're going to die. And the epichaperome negative or low cells, when treated with PUH71, are going to survive. We have validated this data by using in vitro assays and in vivo assays. So shown in here, you can see that we see a correlation between the percent survival and the relative abundance of this epichaperome. This is all in primary AML samples. We also confirmed this using cell lines of different tumor types, and the concept is still valid. In addition, we confirmed this in vivo using primary derived xenografts with different levels of epichaperome abundance. And as you can see here, the more abundant were the cells to epichaperome inhibition, or the epichaperome, they were more sensitive to treatment with the epichaperome inhibitor PUH71 in vivo. In addition, we're showing here the levels of hyperactivated signaling, as you can see, measured by phospho-S6 and phosphostat-5. And you can see the more phosphorylated networks they have, the more abundant epichaperome they had, the more sensitive to PUH71 they were. It is important to mention, as my laboratory focuses on leukemia stem cells, which are the cells that give rise and maintain disease, and the best way to detect leukemia stem cells is doing serial transplantation, we needed to see if we were able to eliminate leukemia stem cells, and that if this assay will be able to predict which leukemia stem cells will be more dependent to this epichaperome inhibition. And as you can see here, an example, AML4, shown here, has very high levels of epichaperome, high levels of signaling networks. Those cells, when transplanted into a secondary animal, were unable to recapitulate disease in these mice, suggesting that we were able to eliminate leukemia stem cells in these animals. In contrast, for example, AML9, which had a partial response to PUH71 in vitro, shown in here, around 20%. When the surviving cells were transplanted into a secondary animal, you can see that the levels of disease were actually higher, suggesting that what we were killing were probably leukemic blasts, and we were leaving behind the leukemia stem cells, and therefore increasing the engraftment on the secondary animals. So we're increasing the abundance or frequency of leukemia stem cells. This clearly emphasizes that it's very important to guide therapies because we could inadvertently enrich for leukemia stem cells, for example. All of this was extremely nice and exciting in the lab. We published actually this paper in 2016, but it was really hard to prove whether or not the big question would be, can we use this type of assays, the PUFITC assay, to actually identify patients and treat patients specifically guided by this drug. So we were very fortunate to start collaborating with the Englander Institute of Precision Medicine on this particular project. And this is a case of a patient that we refer as PM254. So this is a very interesting case because it is a female, 61-year-old woman that was diagnosed with an accelerated phase myeloproliferative neoplasm back in 2013. So what was really interesting about this patient is that she did not present the typical mutations that are related with an MPN. So clinically, the features of her disease looked like an MPN, but she was negative for all the mutations that are commonly found for MPNs. And they are listed here. She was negative for BCR-ABL, PDGFRA, alpha and beta, FGFR1, JAK2, et cetera. So she was negative for anything that will tell you is an MPN and how to treat her. Because she kept progressing with her MPN, she went to an unrelated donor allogeneic bone marrow transplantation in 2014. However, she relapsed from her transplant and in 2016, and she began treatment with hydroxyurea just to keep her counts lower. She also presented a typical graft versus host disease, which was just in one toe, which then led to treatment with ruxolitinib. And this is going to be relevant for later on when I mention the combination treatment. So in 2017, a bone marrow biopsy was taken from her and showed that she was progressing to AML with fibrosis. And she started developing splenomegaly and losing weight, and she was just progressing very rapidly on AML and aggressive disease, and she was not responding to any available treatment. So she was referred to the Eipm for sequencing, and she was sequenced, and what was a very interesting finding, that she had a very novel translocation that has not been found in any other patients so far. And this consists in the translocation of PML with SIG. What we found, so initially, we wanted to determine what this translocation would be doing. However, you have to think that this patient was in an urgent need of treatment, so we needed to also know how can we treat this patient. So when we're trying to define what options we could have, SIG is a tyrosine kinase, that there is actually an inhibitor against SIG. So we start looking for phosphorylation of SIG. So in the lab, my postdoc, Mayumi Sugita, decided to test every single phosphospecific antibody we had in the lab. In addition to SIG, phosphostat, phospho-6, et cetera, you name it, she tested them all. And she found that this patient had hyperactivated signaling. So every single phosphospecific antibody we tested, it was positive for her. That led us to think that maybe she would have the epichaperone, high epichaperone abundance, since she had hyperactivated signaling networks. In order to maintain this high demand of signaling, maybe she would have also an addiction to the epichaperone. So what we did is we use our assay, the PU-FITC assay, in the cells of this patient. So here I just want to show you that we sorted lymphocytes, blasts, and granulocytes in order to determine where the translocation was present, and shown here is in the blast and in the granulocytes, but not on the lymphocytes. What is important to mention about the lymphocytes is that lymphocytes are donor-derived, so that's the only thing this patient has left from her transplant. So here you can see that the lymphocytes are negative for the epichaperone. The blast and the granulocytes are positive for the epichaperone. Because the positivity, we then went to test the in vitro sensitivity of her cells to PUH71, which is the epichaperone inhibitor. So on the top, you see the patient cells. On the bottom, you see the control cell line. You can see here that the response of the patient cells was very similar to the most sensitive leukemia cell line we have in the laboratory. Because of this, Dr. Gail Robos, from the director of the leukemia program, started a single patient IND. So in a compassionate trial, we reached to the company and we started treating this patient with PUH71. The interesting thing is because we have the PU-FITC probe, we can measure constantly the levels of the epichaperone on this patient. And this is important because, one, we were able to determine we had target engagement, so meaning that after the patient started treatment, the epichaperone levels that we were able to detect decreased. And you can see here that over time, those levels were going down. You can also see very rapidly that her whole white blood cell counts decreased and became normal. And her blast count also decreased to very low levels, to the point that at day 42, she achieved complete remission. Until till now, today, she's still in complete remission and living pretty much a normal life. Here you can see that after time of treatment, we also saw a decrease in signaling networks. In this case, we're showing phospho-S6 kinase. So you can see that was also achieved. Importantly, we also look at the stem cell populations, and you can see here that as she began treatment, you can see a very clear decrease over time of the phosphorylation of S6 kinase. And sorry, you can see it here at day 11, it's decreased, and the way we determine leukemia stem cells is by the phenotype CD34 positive, CD38 negative, CD123 positive. So the most interesting thing after this that we found was that when we start looking at the immune cells, we also saw that this patient had what we called an increase of pro-inflammatory monocytes and intermediate monocytes. So all of these monocytes have an effect, potentially immunosuppressive effect. So over time, these cells were normalizing, and importantly, the T cell populations were totally not affected. So we've been monitoring this patient since she started treatment. So every time she comes to the clinic to receive a treatment, around every 3 weeks, we monitor her tumor cells, if there is any tumor cells coming coming up, and her immune cells. So an interesting thing we've been observing is that over time, if we see a blip or a tiny amount of tumor cells coming up, let's say 1% tumor cells in the peripheral blood, these tumor cells have high levels of epichaperone. But what we found, what was interesting, before these tumor cells come up, we see these pro-inflammatory monocytes start to appear. As the pro-inflammatory monocytes start to appear, they express PD-L1, and then the lymphocytes that are donor-derived start expressing high levels of PD-1. The epichaperone is also very high in these pro-inflammatory monocytes, so as soon as we start treatment again, these pro-inflammatory monocytes go away, then the tumor cells go away, and then the T cells stop or decrease the expression of PD-1. So we think that the part of the success of this patient has to do with also a role of the epichaperone in the immune cells and in the immune suppressive microenvironment. So it's decreasing this immune suppressive microenvironment, which is helping this patient to achieve this very long-term remission. So as a summary, I discussed with you that we have a very novel flow cytometry-based tool that we're using to evaluate epichaperone at the single cell level and help us identify this patient that was predicted to be sensitive to PUH71. This patient was a very poor prognosis patient that relapsed even after allogeneic bone marrow stem cell transplantation, and she's still in remission. So we're currently screening more patients, and we are in the way to treating more patients. And just finally, just to discuss something that also emerged from this collaboration with the Eipm, is we're trying to implement the epichaperone abundance assay in the organoid platform. So we've been working with the Eipm in order to use this tool to be able to distinguish the epichaperone high and the epichaperone low organoids in order to predict also responses to this drug. And so far, it looks promising. We need to do some tweaking, but that's something that we're looking forward to do with the Eipm. So just to finalize, it's a all hands on deck effort, so that's why I don't have a particular picture, because it involves a lot of people, and most of them are, you can see them here listed. I think when we are writing and preparing this paper, it's going to be a paper with more than 25 authors and one patient. But we are extremely happy, extremely encouraged. We have met the patient, and it's an effort that opened doors for us for using this epichaperone assay and to bring forward these epichaperone therapies using a guided method. So thank you.
Thank you for a wonderful talk. I think in the interest of time, we'll go to the coffee break and talk. And our last speaker for the session is Dr. Marcin Emlinsky, who will discuss about novel patterns of complex rearrangements across thousands of cancer genome grafts.
Okay. My lab's interested in complex structural variation or genomic rearrangements in cancer, and the reason for that is there's a long history of genomic rearrangements leading to some of the most famous targetable oncogenic gene fusions. More recently, there's been an interest in how genomic rearrangements can land outside of coding regions and rewire chromatin and epigenetic states, and in certain rare cancers has been shown to drive tumorigenesis through something called enhancer hijacking. And then a third feature of these complex variants that motivates our interest is the study of mutational processes that shape cancer genomes and both serve as a readout of DNA repair defects and potential exposures to mutagens. And I got interested in genomic rearrangements when we did some of the first whole genome sequencing studies of lung adenocarcinoma, which a significant fraction of these patients are never smokers. And shown here are two circus plots that are one way to summarize the genome-wide rearrangement state of these tumors, where the middle shows the junctions that are connecting regions that were previously distant, and the periphery shows the copy number alterations. Since making this figure, I've forgotten which patient is which, and that really underscores the fact that we still know very little about what's driving genomic rearrangements, because if we looked at the single nucleotide variants in these patients, we would immediately be able to tell which was the smoker. So we've been involved in a international consortium in analyzing large compendia of cancer genomics data with the ICGC, PCOG, and TCGA. And this is just a snapshot from a preprint of the papers are in press currently, looking at the spectrum of rearrangement burden, which is quantified here by the number of rearrangement junctions per patient. And you can see that there's 2 orders of magnitude difference between the least rearranged leukemias and myeloproliferative neoplasms, thyroid cancers, and the most rearranged sarcomas, lung adenocarcinomas, and squamous cell cancers, breast and ovarian tumors. Where we want to get to is sort of the mature state that the field of SNV signatures has gotten to, where there's distinct footprints of aging, of exposure to specific mutagens like tobacco smoke or UV light, DNA repair defects, and even endogenous mutagens like this APOBEC signature. And here, of course, the problem's a bit easier because all you need to do is look at the trinucleotide context where the mutations land, and then there's a lot of those mutations, over 2 orders of magnitude more than there are rearrangements. One fundamental problem is we don't actually know how to count rearrangement events. The atomic unit of rearrangement detection in short-read sequencing is the rearrangement junction, which is just a pair of locations that are distant in the reference genome but have been brought together through a rearrangement event. And there's 2 locations, and each of them have 1 of 2 orientations, so that gives you 4 possibilities. But when we think of rearrangement events, we're thinking of a actual sort of evolutionary step in a genome, and a lot of our lexicon is borrowed from constitutional genetics, where the variation is a lot more simple. So here you'll have certain rearrangements that are like deletions and duplications, which involve a drop in copy number, a gain in copy number, and a single junction. And then you could have these 2-junction rearrangements like translocations and inversions that don't have a footprint in copy number, but involve 2 junctions. Now in cancers, things get a lot more complex. Rearrangements can be stacked on top of each other, and even simple rearrangements can give you complex patterns. And the other problem is that we have an evolving cancer genome whose coordinates we don't actually know. So if we project these rearranged alleles, in this case one that underwent 2 tandem duplications that are nested in one another, the second tandem duplication duplicated the first, you can get this complex allele that, unless we know its phase, we're not going to necessarily be able to infer what happened to that locus. And an event here that looks like a deletion actually is a duplication. It's a late duplication. So really the context of these rearrangements matters, and we need a more integrative approach to study these cancer genomes. And of course, that's just the tip of the iceberg in cancer genomics. Chromosomes shatter. They undergo these chromathripsis. They undergo these multi-way balanced rearrangements that involve several loci. Breakage fusion bridge cycles that give you these high-level amplifications through these clusters of foldback inversions that are the result of end-to-end fusions that give you these palindromic sort of paper folding patterns when you project the loci onto the reference genome. And then we have these really fascinating spaghetti soups that my lab is essentially working to characterize. So we've developed novel computational methods that also come along with novel visualizations that we're using to browse these genomes and sort of with this new interactive genome browser, a nonlinear sort of graph genome browser. And the nodes in this graph are the intervals of the genome, and the height of the plot shows their copy number. And in addition, we're connecting these loci with these rearrangement junctions to which we also assign copy number. And this is one type of complex rearrangement event called a templated insertion chain, where if all these junctions are in cis, essentially this involves some kind of complex mutational process that brings together many distant loci in the genome into a single cis allele. Deriving these graphs isn't totally trivial, and we've basically worked very hard to build this computational pipeline that allows us to take the raw input, which we can just summarize here with this circus plot, which is this high density read depth of sequencing read depth along the genome, and that's in this periphery, and these rearrangement junctions that are in the center. And these are two signals of a single cancer genome structure, and they're usually analyzed completely separately. So we know, though, intuitively that when a region of the genome is amplified, that region, unless it's at the end of a chromosome, needs to have a left and a right neighbor. So there's just a fundamental sort of physical consistency that we expect in cancer genome structure, and basically no algorithm considers that. So we've developed this process that takes this sort of high density read depth, noisy read depth data and infers this graph where we have both the junctions and the nodes are assigned a copy number. And in certain cases, we might not be able to find a junction that explains an obvious copy change, and that's pretty expected because in certain cases we might, our sequencing platform, short read sequencing, may be blind to certain parts of the genome that lack, that are repetitive, and so we want our model to be robust to this. And so we built this pipeline and thoroughly benchmarked it, and it's really just a matter of quantitatively bookkeeping these copy changes, and we do this with something called a mixed integer quadratic program, which kind of tries to fit this read depth while minimizing the number of loose ends that we fit. And I'm not going to go through the benchmarking that we've done against the handful of tools that have been developed for this, but the main thing is what do we get out of these models? In addition to getting the copy number of every interval, we actually get the copy number of every junction. So what does that mean? When you have a one copy junction in a low copy region, well, we have one, that means that we predict that there's one copy of that interval that's fused to some distant part of the genome, and then two copies that are unrearranged. And there's various configurations of these, and what I want to argue in this talk is that this is an important feature of cancer genome structure that we can use to discover recurrent patterns. And we can infer this junction copy number better than other tools, and this is data from simulation, but we can actually also validate this using 10x chromium linked read whole genome sequencing, which gives us actually a more direct readout of junction copy number, but very few genomes have been sequenced with linked read technology, though my lab is also working on this. But here you can see that our predictions from short read whole genome sequencing is that these are two low copy junctions, so then as predicted, this off diagonal barcode intensity is going to be smaller than for a high copy junction that's present at 10 copies. So our short read whole genome sequencing predictions, for which we have many tumors sequenced, are very concordant with what we would get if we actually ended up doing linked read sequencing. When we combine our inferences on the interval and junction copy number, we get more faithful reconstruction of these loci. So this is JABA, our algorithm, compared to other tools that attempt to build these graphs. And here you can see there's a bunch of fold-back inversions that explain the sort of high copy number that this locus achieved. And more broadly, we can now take these graphs and analyze them to de novo diagnose previously identified complex rearrangement patterns like chromothripsis, chromosome shattering, these multi-way balanced rearrangements, and these templated insertion chains. In addition, we can use this graph because we know the actual genome in a cell is not a graph. It's the superimposition of several linear or even circular alleles, but we can take these graphs and actually try to deconvolve them into alleles. So these alleles here, you can see are, when you sum them together, you're going to get this graph in terms of there being two copies of this locus and then one copy of this junction, et cetera. And these linked reads will help us sort of disambiguate two consistent solutions that are consistent with a short read whole genome sequencing data. But of course, again, we don't have thousands of whole genome sequenced with linked reads or long reads yet, but we do have thousands of whole genome sequenced with short read whole genome sequencing. So we've taken a large compendium of these data, some of which have been generated at the Eipm, including roughly 400 precision medicine cases, roughly, as well as additional cohorts that have 1,800 samples that have not yet been analyzed in pan-cancer studies. So this is the largest study of pan-cancer whole genome sequencing structural variation that will be published and will be larger than the study that the PCOG is going to be producing. So one thing that we can see in sort of validating our approach for junction copy number inference is that we get a close correlation between the read depth difference at segments and this junction copy number. And the distribution of junction copy number roughly has two modes. One is these junctions that have low copy, which is like less than 2 or 3 copies, and this high junction copy number, which we sort of draw by looking at this histogram. And examining the patterns of these low and high copy junctions, we can start to look at understanding the landscape of complex structural variation across these 3,000 cancer genomes spanning 32 cancer types. So one thing that we notice when we focus on these low copy rearrangement junctions is that when they form clusters, they tend to be quite pure in either deletions or duplications. And you can see that the low copy junctions are quite enriched in these deletion pure or duplication pure rearrangement clusters. When we quantitatively look at the clustering of these low copy duplications in individual patient samples, we can build a background model that helps us nominate these towers of tandem duplications, which we call Pirgo. So if you follow cancer genomics, there's a kind of a goofy trend to name things with Greek, using Greek names, and we have a couple of Greek members on our team. This is named after these Pirgo in the island of Santorini. And you can see that these tandem duplication towers are distinct from the patterns that we see when we see these isolated simple duplications. And these Pirgo cluster in ovarian, endometrial cancer, esophageal, and breast adenocarcinoma. The deletion patterns, low junction copy deletion patterns show an even more striking clustering of these pure deletion clusters, which we call rigma, or ditches or rifts in these cancer genomes. And you can see here that we have a set of overlapping deletions, which are in this locus that's about 100, few megabases wide, and the lesion itself is about a couple of 100 kb. And these are incredibly enriched in Barrett's esophagus, which are 320 of our whole genomes are from Barrett's esophagus, as well as esophageal adenocarcinoma, and are relatively rare but not infrequent in other cancer types. And this is in contrast to the pattern that we see with a simple deletion. Rigma have a distinct distribution along the genome, so you can see here that when we look at reference annotations of replication timing, they're really enriched in late replicating regions, and these are the kind of p-values that I like to report. And they're also enriched quantitatively, but not exclusively, in what have previously been annotated as genomic fragile sites. Rigma also tend to land in these multi-megabase genes, which is really interesting because if you put this together, we think that this is a signature for genomic fragile sites. How would these form? Well, one hypothesis would be that these long genes take longer than a single cell cycle to transcribe, and as a result, they collide with the replication machinery, and replication transcription collisions may be responsible for this pattern. Here are examples just to show you how visually recurrent this pattern is. These are totally independent patients. The row one, a few of these are Barrett's esophagus. A few of these are, actually this column is Barrett's esophagus. This column is esophageal adenocarcinomas, mislabeled. But you can see that these are roughly, again, all of these land in FHIT, which is a known site of frequent deletion in esophageal adenocarcinoma. And you can see they actually target this middle region of FHIT, and the pattern is quite recurrent. So what's interesting is when we plug in all those covariates of the rigma density, we still get hotspots. So it kind of raises the question of whether these hotspots are drivers, or they're still sort of just passengers that are cell of origin tattoos. And I think that's an interesting pattern to look at. So rigma are not chromothripsis. Chromothripsis is this whole chromosome shattering. When we look at the patterns of chromothripsis along our samples, the size range of chromothripses are between 10 and 100 megabases as expected, but these rigma are quite short. They're around 1 megabase on average. Another interesting thing is that when we try to phase these these events. And of course, we can only do that with limited confidence because it's short read data, but we can ask how many of these junctions could we place in cis for a given rearrangement event. And for chromothripsis, we can usually place most of the junctions into a single allele, which means that we think there's a single derivative allele that carries the sort of footprint of that event, which is consistent with the fact that chromothripsis is probably a catastrophic event that occurred at a single point in time. But with rigma, we can rarely phase the junctions in cis, which means that they likely occurred in trans. So when we validated this by doing some 10x chromium linked read sequencing, which gives us sort of these 50 kb fragments with short reads, we can use the barcode association to phase these alleles. Indeed, we take a rigma, this is one in a small cell lung cancer, just showing that these aren't exclusive to esophageal adenocarcinomas, and we can find these alleles that are in parallel, sort of summing up to this locus, and you can see that each of them have a separate combination of these junctions in trans. And so that makes us think that this is the result of a gradual mutational process that adds deletions to these fragile sites. And indeed, when we look at our Barrett's esophagus samples, these are pre-malignant lesions that are predating, they're not cancer, they're predating esophageal adenocarcinoma. When we look at the patterns of rigma in these, so we have 4 samples from each patient, and actually at 2 time points. The rigma tend to be shared by all samples that have been obtained from that patient, like roughly 70% of patients will have, or 70% of rigma will be shared by all 4 samples, while the deletions tend to be more private. When we analyze one of these Barrett's esophagus lesions, we can actually see, though, that even though every sample in that patient shares a rigma, meaning that there is a rigma at that location, they are accumulating deletions. So these are kind of loci that are basically being sort of recurrently deleted, and even alleles that were previously deleted, we can see evidence of those alleles acquiring additional deletions. Pirgo cluster in super enhancers, and actually we can find the most recurrent site of Pirgo is the super enhancer of MYC, and MYC is a known oncogene. We can see here are examples of MYC Pirgo in lung and squamous cell cancer. And what's interesting about these Pirgo, first of all, they're on a really large scale, so actually 10x chromium is sort of useless for these, but what we've been using is bio-nano optical mapping, which gives us much longer fragments and allows us to directly visualize the order of molecules. And sort of deconstructing this short read whole genome sequencing graph of these tandem duplication towers, we surmise that these loci, unlike rigma, put all of these duplication junctions in cis. So it's really interesting if we combine that with the knowledge that these are clustering and super enhancers, could this be a mechanism for cancer cells to bring together, let's say, not just duplicate a super enhancer, but bring many super enhancers sort of in tandem and create a really powerful signal to upregulate gene expression. We've also analyzed the clusters, so that was looking at the low copy junction patterns. Here we looked at what are the reasons that tumors will, or mechanisms by which tumors amplify genes to a high copy number. And we can, again, take this graph-based approach where we look at subgraphs of these cancer genomes across these roughly 3,000 graphs that, and the subgraphs that contain amplified regions, and identify those that have a high copy junction and annotate these subgraphs based on a few features, some of them based on the fold-back inversion status, the maximum copy number, the maximum junction copy number, and the total number of high copy junctions in those regions. And that gives us 3 categories when we sort of, in an unbiased way, cluster these regions just based on these few features. One category appears to be these double minutes, which are sort of high amplitude regions. This is a mesothelioma that amplified this region to almost 180 copies. And so this is likely extrachromosomal DNA. BFB cycles can reach high amplitudes as well, but through these clusters of fold-back inversions. And then a third pattern that we call that resembles a typhoon, which we call typhonus. And these are clusters that have a really high number of high copy junctions, but not just high copy junctions of any sort, these high copy fold-back inversions. And these can stretch, you can see here the scale is 10 megabases for really large lengths. So actually these typhonus are much larger than BFB cycles or double minutes, though we can't directly measure the size of these loci, but we estimate that their size would be somewhere between 100 megabases and a gigabase in length. Of course, that's a conjecture, but they don't give you higher copy numbers than these other mechanisms, but they actually span much larger chunks of the genome. And so unlike BFB cycles and double minutes, these cluster in sarcomas and melanomas. And when we zoom in on sarcomas and melanomas, we see that 80% of dedifferentiated liposarcomas carry a Tiphonis, while it's very rare in other types of sarcoma. And this is a really interesting observation because in melanoma you have non-cutaneous melanoma, and acral melanoma is a subtype that's actually equally responsive to immunotherapy as cutaneous melanoma, but has no UV mutations. So it's sort of a mystery as to how do these acral melanomas become immunogenic? How do they generate neoantigens? Well, we see an incredible enrichment of these Tiphonis and acral melanomas. So a conjecture is that this high copy, massive high copy amplification event that may generate many high amplitude fusions could be a source of neoantigens. These Tiphonis target known cancer genes like MDM2, CDK4, cyclin D1, a lot of the known players of amplification, but we think it's just one of the sort of grab bag of mechanisms that different cell types have to amplify these oncogenes. So I'm probably going to run out of time, but we see that these Tiphonis are enriched in a mutational process of junction-centric hypermutation, but outside of the apobec context. This hasn't been previously described. Contagious has been sort of described as a process of apobec mutagenesis around rearrangement junctions. So that's a novel sort of finding from this study. And then we can use these new features that we've built through analyzing these graphs to build, to classify tumors into categories based on the rearrangement events that they are accumulating. So every column here is a sort of normalized burden of a particular rearrangement event. And one thing that you see here is that the BFB and Rigma, double minutes, and Pirgo and Tiphonis, these features that we've sort of rigorously can identify in these tumors are independent from the tumors that have chromothripsis or chromoplexy or some of these previously identified events. So we do think we're defining new mutational events that are independent from those that have been previously characterized, and actually those clusters do have specificity in terms of tumor types. So for example, this DDT cluster contains deletions, duplications, and templated insertion chains, and is enriched in BRCA1 loss of function, actually both germline and somatic. We see this esophageal-dominated cluster, a cutaneous melanoma cluster with chromothripsis, and additional genotypic correlations. These clusters that we've defined on the basis of these features are also prognostically important. So this is a cluster that contains BFB cycles and Rigma, and you can see it has a poorer survival. Pirgo clusters have a poorer survival, and as well as clusters harboring or tumors harboring Tiphonis. And when we correct for the additional covariates such as tumor type and age, TMB, even P53 status, these features remain significant. So in conclusion, we've completed the largest study of complex structural variation and identified 3 novel patterns of complex genomic rearrangements using this new paradigm for analyzing whole genome sequences. These Rigma are late replicating rifts or ditches of these trans junctions arising early in esophageal adenocarcinoma tumorigenesis. Pirgo are these super enhancer associated duplication towers, and Tiphonis are likely extrachromosomal, though we can't say, but they're massive events that are enriched in dedifferentiated liposarcoma and acral melanoma, and these allow us to identify prognostically relevant features of the cancer genome. We're exploring these with long-range whole genome sequencing technology and looking in cohorts with patients with inherited DNA repair defects, and then just want to acknowledge the members of my team that contributed to this work and the funding. Thanks.
We'll catch up on the coffee break. Now let's thank all the speakers for a great morning session, and we'll have a 10 minutes coffee break, and we'll be back for or panel 1 on digital technologies for clinical care. So 10 minutes. Thanks.